In June of 1984, a 32-year-old doctor in Perth, Australia, walked into his hospital laboratory, picked up a flask of bacterial broth, and drank it. The broth contained billions of live Helicobacter pylori organisms, spiral-shaped bacteria that nobody in medicine believed could survive in the human stomach. Within 3 days, he was vomiting. Within a week, his breath smelled so foul that his wife told him she could identify him from across the room with her eyes closed. Within 2 weeks, a biopsy confirmed what he had expected. His stomach lining was inflamed, colonized, and actively infected. He cured himself with a course of antibiotics and bismuth. Then he wrote it up as a paper. Almost nobody cared. His name was Barry Marshall. The man whose bacteria he had confirmed, the pathologist who first spotted them, was Robin Warren, a quiet, meticulous staff pathologist at Royal Perth Hospital. Warren had been looking at stomach biopsies since 1979, and he kept seeing the same thing. Small, curved, spiral bacteria nestled in the mucus layer of the gastric lining. They were always there, not occasionally, not in a handful of samples. In slide after slide, patient after patient, the same organisms appeared in the same location, hugging the epithelial cells just beneath the mucus. He flagged them in his reports. He circled them on slides. He showed them to colleagues. The standard response was that bacteria could not survive in the stomach. The pH of gastric acid sits around 1.5, roughly the same acidity as battery fluid. Textbooks were explicit. The stomach was a sterile environment. What Warren was seeing, they told him, was contamination, artifact, noise. He kept looking anyway. By 1981, he had documented the bacteria in over 100 biopsies. The correlation with inflamed tissue was overwhelming. The explanation from his department remained the same: impossible. Marshall was a registrar in internal medicine, junior, ambitious, and willing to listen.
When he met Warren in 1981, the partnership made immediate sense. Warren had the observation. Marshall had the energy and the clinical access to pursue it. They began collecting biopsy samples from patients with gastritis and peptic ulcers, culturing the tissue, and waiting. The standard incubation period for bacterial cultures was 48 hours. After 2 days, the plates showed nothing. Again and again, nothing grew. Then came Easter weekend 1982. The lab technicians went home for the holiday. A batch of culture plates, inoculated with biopsy tissue from a patient with chronic gastritis, sat in the incubator, scheduled for the standard 48-hour check. Nobody was there to check them. The plates sat for 5 days. When the staff returned on Tuesday morning, something had changed. The agar surface was dotted with small, translucent colonies. The bacteria had grown. They had simply needed more time than the protocol allowed, longer than any known gut pathogen. Longer than anyone had thought to wait. Marshall stained the colonies under a microscope. The same curved, spiral shape Warren had been circling on biopsies for 3 years. Alive, culturable, real. It was not a breakthrough born of genius. It was a breakthrough born of a long weekend and an empty lab. Marshall later called it the luckiest accident of his career. The bacteria would eventually be named Campylobacter pyloritis, then renamed twice more before settling into Helicobacter pylori, a name that would appear on a Nobel citation 2 decades later. By 1983, they had enough data to present a hypothesis. Stomach ulcers were not caused by stress, spicy food, or excess acid. They were caused by a bacterial infection. The bacterium was Helicobacter pylori. It could be killed with antibiotics. The cure was cheap, fast, and permanent. The medical world was not impressed. The stress model had weight behind it. In the 1950s, psychoanalysts had classified peptic ulcer as one of the holy 7 psychosomatic disorders, conditions believed to originate in the mind. The ulcer patient had a profile: ambitious, anxious, male, overworked. Treatment reflected the theory. Patients were told to avoid spicy food, reduce anxiety, take antacids, and, in severe cases, submit to surgery. Partial gastrectomy, the removal of the lower portion of the stomach, was a routine procedure in teaching hospitals through the 1970s.
Scalpel handed off.
The theory was coherent, the treatment was profitable, and the patients kept coming back. Nobody questioned whether the model was wrong, because nobody had reason to.
The system worked for everyone except the patient. In June 1984, Marshall and Warren published their findings in the Lancet. The paper laid out the evidence. The bacterium was present in the stomach lining of virtually every patient with gastric ulcers and most patients with duodenal ulcers.
Koch's postulates, the gold standard for proving that a specific organism causes a specific disease, had been partially fulfilled. The remaining step was to demonstrate that introducing the bacterium into a healthy host caused the disease. Marshall could not get ethics approval to test this on volunteers, so he tested it on the only person whose consent he did not need. He did not tell the hospital ethics board. He told his wife, but only after he had already done it. The preparation was clinical. He fasted overnight, had a colleague perform a baseline endoscopy to confirm his stomach was healthy, and then swallowed approximately 1 billion H. pylori organisms suspended in alkaline peptone broth. The taste, he later recalled, was unremarkable. The effects were not. By day 3, he was nauseated. By day 5, vomiting. By day 8, a second endoscopy showed acute gastritis, the mucosa inflamed, the bacteria visible in biopsy samples taken from his own stomach wall. He had fulfilled Koch's postulates in his own body. He treated himself on day 14 with antibiotics and bismuth subsalicylate. The infection cleared. The gastritis resolved. The experiment was over. 3 months later, at a conference of infectious disease specialists in Brussels, Marshall presented the results. He showed the endoscopy photographs, his own stomach lining before and after infection. He showed the culture plates. He showed the biopsy slides with the spiral bacteria colonizing tissue that had been cleaned 2 weeks earlier. The response was dismissive, which was worse than hostility. Gastroenterologists in the audience pointed out that the sample sizes were small, the controls were limited, and the claim was extraordinary. Ulcers were a chronic condition managed with medication. The textbook model had been stable for decades. Stress, diet, acid overproduction. This was the accepted triad. To suggest that a 2-week course of antibiotics could cure what the profession had spent decades treating with acid suppressors, dietary changes, and psychotherapy was, to many in the room, absurd. One attendee reportedly told Marshall that he was too young to understand the complexity of the disease. Marshall was 32. He had infected himself with the organism under discussion. The audience had read about it in a journal. The commercial reality was harder to ignore than the clinical evidence. By the mid-1980s, a drug called cimetidine, marketed as Tagamet, had become the first pharmaceutical product in history to reach $1 billion in annual sales. It worked by suppressing stomach acid production. Patients took it daily. They took it for years. Some took it for the rest of their lives. The business model was elegant. The drug treated the symptom, not the cause. The patient improved. The patient relapsed. The patient refilled the prescription. When Tagamet's patent expired, a second generation of acid suppressors arrived: ranitidine, famotidine, omeprazole, each worth billions more. Omeprazole, sold as Prilosec, became the best-selling drug in the world by the early 1990s. The global market for ulcer management was estimated at $8 billion per year. Marshall was proposing to replace that entire revenue stream with a 2-week course of generic antibiotics costing less than $50. The economics were not ambiguous. A cured patient is a lost customer. The resistance was not a conspiracy. It did not require coordinated suppression or institutional malice. It required only inertia. Textbooks said ulcers were caused by acid and stress. Professors had taught that model for decades. Pharmaceutical companies funded the research that supported it. Insurance systems reimbursed the drugs that treated it. The infrastructure of ulcer medicine was enormous, profitable, and self-reinforcing. The same pattern appears everywhere a profitable myth meets an inconvenient correction. The correction does not need to be suppressed. It needs only to be unfunded. And for roughly 10 years, Marshall's discovery was. During that decade, Marshall struggled to get funding for large-scale replication studies. The scientific community did not reject his data outright. They simply declined to prioritize it. Grant committees questioned the methodology. Journal reviewers asked for larger trials. Conference panels slotted him into afternoon sessions that half the audience had already left. Gastroenterology departments continued to prescribe acid suppressors. Patients continued to take them. Some developed side effects: bone loss from long-term proton pump inhibitor use, kidney problems, nutrient malabsorption. Some relapsed within months of stopping treatment. Some underwent vagotomy, surgical cutting of the vagus nerve to reduce acid secretion, a procedure that carried permanent digestive consequences. The cure existed. It cost 2 weeks of pills. The system was not structured to deliver it. Marshall moved to the University of Virginia in 1986, partly for research opportunity, partly because the Australian medical establishment had made clear he was not welcome in its upper ranks. From Virginia, he continued publishing. The data accumulated. The resistance held. The pattern eventually broke, not because the establishment changed its mind, but because the evidence became too heavy to set aside. Independent researchers in other countries began confirming Marshall and Warren's results. A team in the Netherlands replicated the culture protocol successfully. Epidemiological studies across Japan, China, and South Korea showed the same correlation between H. pylori and ulcer disease. A landmark study in Ireland treated infected patients with antibiotics and tracked relapse rates. They plummeted. By the early 1990s, the data were no longer a hypothesis from a junior doctor in Perth. They were a wall that no amount of institutional inertia could hold back. In February 1994, the National Institutes of Health in the United States issued a consensus statement. Most peptic ulcers were caused by H. pylori infection, and the recommended treatment was antibiotic therapy. The language was definitive: no hedging, no further research needed. The panel included gastroenterologists who had spent their careers prescribing acid suppressors, some of whom had publicly dismissed Marshall's work less than 5 years earlier. The shift was not graceful. There were no apologies. The textbooks were simply rewritten, and the previous edition was quietly retired. 10 years after the Lancet paper. 10 years after the self-experiment. 10 years during which millions of patients had been treated for a disease whose cause was already known and whose cure was already available. The number of unnecessary surgeries performed during that window has never been calculated. The number of patients who spent a decade on daily medication they did not need has never been tallied. The cost of the delay is not theoretical. It is personal, repeated across millions of medical records that will never be audited. The mechanism that drove the delay is instructive, not because it was built from bad data, but because it was sustained by profitable data. Every clinical trial funded by pharmaceutical companies confirmed that acid suppressors worked. They did work. They reduced symptoms. They healed ulcers temporarily. The trials were rigorous, peer-reviewed, and honest. What they did not test was whether the underlying cause could be eliminated entirely. Nobody funded that question because the answer would have destroyed the market. The Nobel Prize in Physiology or Medicine came in October 2005, 21 years after the original publication. By that point, the treatment had already become standard worldwide. H. pylori eradication therapy was in every clinical guideline in every developed country. Warren and Marshall shared the prize. The committee's citation was straightforward: for discovery of Helicobacter pylori and its role in gastritis and peptic ulcer disease. Marshall was 54. Warren was 68. Neither man had become wealthy from the discovery. The antibiotics that cured ulcers were generic, off-patent, and cheap. There was nothing to license. The pharmaceutical industry that had resisted their work for a decade made its money elsewhere. The story should end there. It does not. Helicobacter pylori has lived inside the human stomach for at least 58,000 years. Genetic analysis of strains across continents suggests the bacteria migrated with early humans out of Africa and co-evolved with its host over thousands of generations. By the time Marshall drank his broth, roughly half the world's population carried the organism. Most showed no symptoms. Ulcers and gastric cancer affected a fraction of carriers. The bacterium was ancient, widespread, and for most people, harmless. Since the 1990s, eradication campaigns have driven H. pylori prevalence sharply downward in developed countries. In the United States and Western Europe, infection rates have dropped below 20%. Ulcer rates have fallen with them. But other conditions have risen in the same populations during the same period. Gastroesophageal reflux disease, Barrett's esophagus, esophageal adenocarcinoma, childhood asthma, and certain autoimmune conditions. The correlation is not proof of causation, but the pattern is consistent enough that researchers, including Marshall himself, have begun asking a question that would have been unthinkable in 1984. Whether eradicating H. pylori from entire populations was the right decision. The modern gut health industry has taken Marshall's discovery and built something he did not intend. Probiotics, marketed as tools for gut restoration, immune support, mental clarity, and a dozen other claims, now generate roughly $50 billion a year globally. Most of these products have limited clinical evidence behind them. The phrase gut-brain axis appears in advertisements for yogurt drinks and supplement capsules alike, and the distance between what the research shows and what the marketing promises grows wider every quarter. Marshall's work proved that a specific bacterium caused a specific disease. The industry it spawned sells the idea that bacteria in general are a solution to everything. A man drank a cup of bacteria in a hospital lab in Perth in 1984 and watched himself get sick to prove a point that the medical profession refused to accept. The proof worked. The profession caught up, slowly. The Nobel Committee caught up, eventually. And the industry that fought the cure built a new one from the same science, selling complexity where Marshall had found simplicity. The organism that lived quietly in human stomachs for 58 millennia is disappearing from the developed world. Whether that silence is a cure or a cost is a question nobody has answered yet. Next time, a scientist flew over the largest living structure on Earth and watched it turn white. The government asked him to keep it quiet. The tourism industry had 6 billion reasons to agree.